Welcome to Study Pharma. In this video, we're going to discuss about urea cycle. Urea cycle is the first metabolic cycle that was described by Hans Krebs and Kurt Henseleit. So it is also known as Krebs-Henseleit cycle. The main aim of urea cycle is to convert toxic ammonia into non-toxic urea. Next, I'll give a brief introduction about urea cycle and we'll discuss the pathway in detail. And at the end of this video, I'm going to explain you the tricks to learn urea cycle and to memorize it better. Since the ammonia produced in deamination is extremely toxic, it is immediately removed from circulation and converted to non-toxic metabolite urea. Now, where does the urea cycle take place? Mostly it takes place in the hepatocytes, that is liver cells, and to a smaller extent in the kidneys. Urea synthesis is a 5-step cyclic process with 5 distinct enzymes. The first 2 enzymes are present in mitochondria, while the rest are present in the cytosol. It means that 2 of the steps take place in the mitochondria and the remaining 3 takes place in the cytoplasm. Now let's look at the pathway in detail. The first step is the formation of carbamyl phosphate. Condensation of ammonia with bicarbonate ion results in the formation of carbamyl phosphate with the help of enzyme carbamyl phosphate synthase 1, shortly CPS1, in the mitochondria. The next step is the synthesis of citrulline. The now formed carbamyl phosphate combines with ornithine, resulting in the synthesis of citrulline. In the presence of enzyme ornithine transcarbamylase. Now this step also takes place in the mitochondria. The so formed citrulline is easily permeable to the mitochondrial membrane and hence it diffuses into the cytosol, so the rest of the steps can take place in the cytosol. Let's move on to the third step, which is the synthesis of ARGINO-SUCCINATE. In the cytosol, citrulline combines with the amino acid aspartate and forms ARGINO-SUCCINATE with the help of enzyme ARGINO-SUCCINATE synthase. This step requires ATP, which is hydrolyzed to AMP. Now the fourth step, cleavage of ARGINO-SUCCINATE. With the help of enzyme ARGINO-SUCCINASE or ARGINO-SUCCINATE lyase, arginine and fumarate are produced from ARGINO-SUCCINATE. The fumarate produced then enters the TCA cycle. Now the fifth step is cleavage of arginine. Arginine is cleaved into ornithine and urea under the presence of enzyme arginase. Ornithine is regenerated in this step and urea cycle completes by the formation of urea. Thus, the toxic insoluble ammonia is converted into non-toxic water-soluble excretable urea. Now let's see some of the metabolic disorders of urea cycle. Disorders of this cycle leads to ammonia intoxication. that is by the accumulation of ammonia. Common symptoms of disorders of urea cycle include vomiting in infancy, irritability, lethargy, mental retardation, etc. Well, let's see some of the disorders. First one, hyperammonemia type 1. And it is due to the deficiency of CPS1 or carbamoyl phosphate synthase 1, the enzyme which is used to form carbamoyl phosphate. Next one, hyperammonemia type 2. It is due to the deficiency of ornithine transcarbamylase. It is an X-linked disorder. The third one, citrullinemia. Here, more citrulline is excreted in urine, that is up to 1 to 2 grams per day. And it occurs due to the defect of enzyme ARGINO-SUCCINATE synthase, the enzyme that converts citrulline into ARGINO-SUCCINATE. So there will be accumulation of citrulline and leads to citrullinemia. Now the next disorder, hyperargininemia. It is the high level of arginine due to lack of enzyme arginase. Now let's move on to the interesting part, that is the tricks to memorize the enzymes in molecules of urea cycle. Let's start with the molecules of the urea cycle. I will use a mnemonic for this purpose. ALS can create an amulet for all unicorns. ALS for ornithine, CAN for carbamoyl phosphate, CREATE for citrulline, AND for aspartate, amulet for ARGINO-SUCCINATE, and for fumarate, all for arginine, and unicorns for urea. If you're finding this difficult, you could create your own mnemonic. Let's move on to the enzymes next. So I have made a mnemonic similar to the first one. So it goes as, can owls always admire amulets? CAN for CPS1, carbamoyl phosphate synthase 1, ALS for ornithine transcarbamylase, always for ARGINO-SUCCINATE synthetase, admire for ARGINO-SUCCINATE lyase, amulets for arginase.